Hello, this should be a relatively short presentation just trying to hit the basics of replication, transcription, and translation. Basically how DNA can control the production of proteins. Now, we have to give credit to Watson and Crick, who first proposed the structure and function of DNA. Now, we will say that Rosalind Franklin, a British scientist who was dating one of them, they took some of her research, so she didn't get credit for it, but Watson and Crick and another gentleman got the Nobel Prize later on. So there's always a lot of competition between scientists. Anyways, DNA is controls cell function by serving as a template for protein structure. Getting into a little bit of the nitty-gritty, 3 nucleotides, so a triplet. We also call this a codon, so here's another term that you need to remember, which will code for a specific amino acid. And amino acids are the building blocks of proteins. Remember, when we eat things, what do we eat that has protein in it? So eggs, meat products, beans, and our body eats that protein, breaks it down into small chains of amino acids, and then our body literally builds amino acids into proteins. Like what? Like muscle tissue. And then the next slide I'll try to go back with something you're familiar with when we talk about codons. This should look familiar. The pop beads. And we remember, what are the color ones, what are the white ones, what are the red ones? Now the white ones are the deoxyribose, so these were the pentagons. And so these, anytime you have OS at the end, OSE, it's a sugar. So here's a sugar, and this red one is a phosphate, and this is one of the nitrogen or nitrogenous bases. And the yellow one, if you remember, is guanine. So recall that these 3 parts, the phosphate, the sugar, and the base, these 3 represent one nucleotide. In the previous slide, there's 3 of these in a chain, and that is called a codon or triplet, and that will help to code for specific amino acids. And we recall there's 20 different amino acids, so 3 of these in order will code for specific amino acids. And so you have different types of amino acids in a chain, and then those will code for a specific protein. And then just always review. So the yellow one is guanine, so you should know what this is. What pairs up with guanine? Remember, G and C look similar, so that pink one is cytosine. So cytosine, guanine. And if I had a green one, let's say I have a green one, then what would it match with? So a green one would be thymine, and then it would match with adenine. So AT, and we have GC. Just real basic diagram, replication. So when you replicate something, you just create more of the same thing. Sometimes we call this semi-conservative. So you have 2 strands, the dark blue, 2 strands of the original DNA, and then just like a zipper, this zips down the middle and will then you'll have bases fill in on the other side, and eventually then you have a full zipper or DNA and another one. Splits again, another DNA, another one. So then we have a 4 after 2 rounds of replication. This seems very simple, and then we're going to get into actually how it unzips in how we can actually make more DNA, such as when we go through mitosis or meiosis. So mitosis, just cell growth, making more cells, like you give it a cut or if you're growing. And then meiosis, when you're making gametes or sex cells, like sperm and egg, where then you only get half of the number of chromosomes. Remember 46 chromosomes? So a gamete or sex cells, sometimes we call them haploid because they have half of the chromosomes. So they would have 23 instead of 46 for a normal whole cell. So I found a diagram that will show us the big picture. Everything down to a chromosome. And remember, humans have 46 chromosomes. And then we're going to look inside the chromosome to different strands of DNA and how it's beginning to replicate. Now remember the phases of mitosis and then meiosis. And we have PMAT, the PMAT. So this is in prophase because it's beginning to pair up. So we just have one chromosome, and then when it's in prophase, it's beginning to pair up, prepare to split apart. So it's beginning to duplicate, but it's still stuck together. Two sister chromatids are stuck together by the centromere. And so you're basically duplicating the DNA. It's replicating at this phase. So we have a polymerase here. Remember, ACE at the end is an enzyme, and enzymes tend to break down things. We remember when we were talking about digestion and nutrition, amylase, ACE, breaks down carbohydrates. Protease breaks down proteins. And lipase breaks down lipids and fats. So here we have polymerase, and it's breaking down DNA. So really it's, I guess not breaking down, but uncoupling it. So we have the zipper. So this one is unzipping the DNA. And then we have another polymerase that's coming in and filling in the complementary. So we have just a lot of extra base pairs floating around inside the cell, and then it's just going to match up. So G to a C, an A to a T, a G with a C, and it'll float in. And this polymerase goes through and fills in along just behind this, and then it fills in, and there's another one that will fill in this way. So in the end, during replication, you'll get two new full strands of DNA. This also double checks for errors, so you don't have a mutation. So just kind of reviewing what I talked about this last slide is that, again, DNA polymerase, ACE at the end, an enzyme, it unzips the DNA and adds new complementary bases. So again, an A, then a T, thymine will come in, another A, adenine, another thymine will come in, C, cytosine, the G will come in. So just remember the DNA polymerase enzyme that adds bases, these bases, to the growing DNA strand. And it also acts as a proofreader. It will go, and if a G tries to come in here and attach, it'll stop, back up, and kick that base pair out, and so the correct one, a T, thymine, will come in. So to go and just double check that there are no errors so that you will have no mutations. Now that we have replication down, making new DNA molecules, we look to transcription. Transcribing, if you transcribe something, you're rewriting it. And then translation, just think about you're translating from a different language. So we go DNA to RNA, so messenger RNA. So it's carrying a message from the DNA in the nucleus into the cytoplasm. And then it will be translated, this code, into a protein. And then something that you should be asking yourself is, what proteins are we making? Now we're looking at RNA. So real quick, DNA, deoxyribonucleic acid, first ribonucleic acid. What's the difference between DNA and RNA? RNA is just a one-strand helix, has different sugars, and there's three main types. So we have the TMR, So T transfer RNA, it's needed to bring the correct amino acids to the ribosomes. Remember, the ribosomes looks like little black dots on the endoplasmic reticulum. So remember, the endoplasmic reticulum is an organelle, mini organ, along with ribosomes. And the ribosomes, we remember, make proteins. That's where proteins are made inside the cell. So the messenger RNA, remember, the messenger carries a message from the DNA in the nucleus to the cytoplasm. And then we have R for ribosomal RNA, forms the structure of the ribosomes where the proteins are made. Reviewing RNA again, similar to DNA, except it's just a single strand. The ribose, OSE, anytime you have OS, it's a sugar. So RNA, ribonucleic acid, one strand. It's very similar to DNA, adenine, guanine, cytosine, but it has uracil, U, instead of DNA's thymine, T. So remember, DNA is miles long, and then it'll actually just open up a little piece here, will open up, and it'll transcribe, it'll write one side of the DNA, let's say just this much, and then it will take this information, go out of the nucleus through the pores, and then into the cytoplasm. So transcription literally is taking this, you can think of a DNA document, and writing just a page or a half a page out of this information and then carry it into the cytoplasm. Alright, this looks a little bit intimidating slide, but we're just translating that message that was in the DNA inside the nucleus. It comes out into the cytoplasm and then goes to where proteins are made, which remember is the ribosomes. So here's just a message. That's all it is. Just think about it like a piece of paper. So we just bring this message to the ribosome where proteins are made, and the ribosomal RNA tells the cell which T transfer RNA to bring, because the transfer RNA has this anticodon, so that just means if there's a here, you got to bring a T. So if you got a U, you got to bring a G, C. So that's just the anti or the opposite. So here is we have this transfer RNA. So it has the, again, we see the opposite. So it's starting to translate this message with the complementary base pairs. And each of these complementary base pairs, remember the triplets, the codon, so 3 of these is a codon for, this is an amino acid. And this is a code for another amino acid. So these 3 code for methionine, and these 3 code for asparagine. And remember, there's 20 different amino acids, and so you might have this one, this one, this one. And it's this different amino acids. Remember, amino acids, if you have chains of them, like here's one, then you could have another 5, another 50, and these amino acids link together in a chain and make proteins. So all of the meat products and eggs and beans and things that you ate, proteins got broken down to amino acids. Now we're taking these amino acids and building it into proteins like hair and muscle fiber. So I've just got some questions I want you to review, and you need to be able to answer these questions for the big ginormous test that's coming up. And I do have the answers that will follow. But you should know these now and review it.